Okay, so today actually we have Dr. Heaney from University of Georgia to give a presentation on modeling of blood disease and thrombus, but he have a kind of time conflict because today he started his first class and the class schedule is the same time as our seminar. So he in a short time we cannot change the speaker, so he do a recorded lecture and last night and send me the entire lecture. And the only thing we cannot do today is the Q&A. We cannot ask the question directly to Dr. Heaney, but I will do the presentation by the recorded lecture. And if we have any question to Dr. Heaney, you can send me email of your question, or you can send the email to Dr. Heaney directly. So today, let me start with a quick introduction. So Dr. Heaney is currently an assistant professor in the School of Medical Materials and Biomedical Engineering at the University of Georgia. And Dr. Heaney received his PhD from University of Connecticut in 2015. And Dr. Heaney has developed multi-scale models based on physical laws using different numerical methods, including molecular dynamics, dissipative particle dynamics, and the spectral element method to simulate biological systems. So his work has demonstrated that multi-scale modeling can bridge the gap between the microscopic and the macroscopic physiologic process and provide innovative approach to study key problems in biology, medicine, and biomedical engineering. So today, Dr. Heaney will introduce his recent work on modeling of blood disease and thrombosis. Then I will share my screen. Let me share my screen. I can share the share. So let me start this.
Okay, good morning, everyone. My name is Heaney. I'm currently an assistant professor of research at the School of Chemical, Material, and Biomedical Engineering at the University of Georgia. So today, I'm very glad to be able to present at the complex flow seminar. But unfortunately, because I have to teach a class on Wednesday morning, so I cannot give this presentation in real time. So I have to make this video record beforehand and send it to Dr. Jen Lee to play it during this seminar time. So hopefully next time I can give a real-time presentation so we can have a better interaction with the audience. Also, I want to thank Dr. Jen Lee and also other organizers of this seminar to provide this platform for the researchers in this community to interact with each other and update each other with our recent research discovery. Okay, go back to my talk today. So the title of my presentation is the Computational Modeling of Blood Disease and Thrombosis. So first, I want to give you a very brief introduction on my research background. So my research interest is multi-scale modeling in the biomedical engineering. So the figure on the right basically summarizes some of my previous work. So from this figure, you can see that by using computational modeling, we actually can simulate the biological process that spans 7 orders of magnitude in the length scale. For example, at the nanometer scale, we can simulate the polymerization of a sickle hemoglobin into these very stiff fibers that can change or alter the morphology of the red cell and also makes the cell stiffer. So this is considered as the root cause of the sickle cell disease. And also we can simulate the protein diffusion in the lipid membrane And at the cellular level, we can simulate a single red cell passing through a very narrow slit and simulate the drastic deformation during his passage of this slit. And also we can simulate the blood flow in the macro vessels and simulate the spleen filtration function at the organ level. And also we can make predictive modeling of this thrombosis in the aortic dissection in both the mouse model and also human being. So the motivation of making all this computational modeling is that first we can simulate some biological process that cannot be observed from the experiment, even either in vivo or in vitro. Also, we can use computational models to examine existing hypotheses for human physiology and pathology of human diseases. And also we can derive a new hypothesis to steer new experiments and new simulations. Second, we can use computational modeling to dissect very complex biological process that always involve many factors. And using computational modeling, we can quantify the contribution of each factor. By doing that, we can identify the critical factor that determine or dictate the critical events, such as vessel occlusions and thrombosis. And also we can make some predictive modeling to help a physician to make a prognosis. And also we can use those computational models to cross-validate so experimental findings, such as in vivo from the in vivo experiment or even in vitro experiment. So next, I think I'm going to use two examples to demonstrate how we can use this computational model to answer some critical biomedical questions. So first, we want to introduce you this spleen function in the sickle cell disease. It's part of our ongoing NIH project, collaborate with Professor Karniadakis at Brown University. So first, a very brief introduction on the human spleen. So human spleen is the largest organ in the lymphatic system. It's actually located on the left side of the abdomen, right above the stomach. So the spleen mainly consists of the two compartments, the white pulp and the red pulp. The white pulp tissue consists of immune cells like the T cells and B cells. So its function is to fight virus, bacteria. And the red pulp tissues basically has a function of filtering the blood to remove diseased and aged red cells. So here is a sketch showing the structure of the human spleen. So you can see that when the blood flow into the spleen, it will first go through this white pulp, where the blood vessels are surrounded by the immune cells, such as the T cell and the B cell, to remove all the pathogens, disease, and bacteria, virus in the blood. So after the blood passes through the white pulp, it's going to flow into the red pulp. And first, it's going to split into two pathways. So the first pathway consists of 80 to 90% of the blood. It goes through this so-called closed circulation and return to the vein. And the rest, 10 to 20%, they have to flow into the red pulp and to be filtered in the red pulp. So here is the two images showing that two red cells are being filtered in the red pulp. So we can see that in order to pass the red pulp, the red cell has to under this drastic deformation. So if there's some compromise on the red cell deformability, so they are going to be retained in the red pulp and removed from the circulation. So here's another sketch showing the summary of the spleen filtration function for the red cell in the red pulp. So in the red pulp, once the blood flows into the red pulp, it's going to first interact with the splenic macrophage. So the macrophage has a function of scanning the surface membrane protein alteration. So if there's any protein defect or protein alteration on the red cell membrane, they are going to be sensed and captured by the macrophage and eventually phagocytosed by the macrophage. So if the red cell can pass or can escape from the scanning of this macrophage, before they return to the circulation, they need to squeeze through this very narrow slit between the endothelial cells on the sinus wall. So this is basically a mechanical check of the right cell. So the right cell with compromised deformability, they will be retained and eventually phagocytosed by the macrophage. So although the function of the spleen in filtering the aged right cell has been well studied, but actually the role of the IES, or the interendothelial cell slit, in filtering the right cell with a significant membrane protein defect in the blood disease, such as the Spherocytosis and Elliptocytosis, has been not well addressed. Also, how the spleen facilitates the maturation of young red cells or reticulocytes has not been investigated in sufficient details. So that's why we perform this computational modeling, use a detailed red blood cell membrane model to simulate the process of the red cell passing through this interendothelial cell slits. So first, I want to introduce you this red blood cell membrane structure. The red cell membrane consists of two components, the lipid bilayer and the cytoskeleton. So the lipid bilayer behaves like a fluid with the transmembrane proteins like the BAN3 and actin flowing in the membrane. And on top of this lipid membrane, there's a cytoskeleton attached to the lipid membrane through the three major binding sites. So there are actin binding site, also BAN3 binding site. And actually, the cytoskeleton is responsible for the mechanical property of the red cell. And also, the lipid bilayer is responsible for the viscous property of the red cell membrane. So to simulate the red cell membrane, we use the open RBC model, where we simulate both the lipid bilayer and the cytoskeleton explicitly. So here, the red particle represents the lipid bilayer, and the green particle represents the spectrin filament. So they are connected at the actin junction, forming this triangular structure. And also, the cytoskeleton is connected to the lipid bilayer at two binding sites. One is at the actin junction, and the other one is at the BAN3 binding site, located at the center of the spectrin filament. And we are simulating this red cell passing through a very narrow slit with the size about 4 micron wide, height is 1.2 micron, and thickness is 1.89. So this size is comparable physiologically to the slit size in the human spleen. So first, we test healthy red cell passing through the slit, and we simulate a red cell with a surface area of 140 micrometer square and a volume of 90 micrometer cube. First, we simulate the red cell passing through the slit, driven by a pressure gradient is 3 Pascal per micrometer, and we find that this driving force is not sufficient to drive the red cell through the slit. And then we increase the pressure gradient to 5 Pascal per micrometer, and we find that the red cell can pass through. And in the next case, we simulate an aged red cell passing through the slit. So the feature of the aged red cell is a reduced surface volume ratio. So in this case, we reduce the surface area of the red cell from 140 to 110, and we find that actually this aged red cell cannot pass through the slit because of this reduced surface volume ratio that compromise the deformability of the red cell. Then we actually increase the driving pressure for this case, and we can see that if we use a larger force, we can pull this aged red cell through this slit. But actually, we find that it did not push the red cell through. Instead, it actually caused the lysis of the cell. So if we zoom in this lysis process, so we can see that when we apply a very large force on this passing red cell, we can see that if we plot this surface area expansion ratio, we can see that the area expansion, well, it is initiated on this upstream part of this red cell, and it's going to accumulate at a certain point, and eventually it's going to create a pore here. And then the pore is going to expand and eventually cause the lysis of the cell. And then next, we simulate a young red cell passing through the slit. So young red cell is actually featured with the redundant surface area. So if we look at it from scanning electron microscopy, it actually show up like a red cell with a lot of wrinkles due to this redundant surface area. And then based on this observation, we actually reconstruct this reticulocyte and simulate it passing through the slit. And we find that after this reticulocyte pass through, it actually can remove the redundant surface area through this vasculation process. And also we note that this red cell transfer to a biconcave shape, which is a typical shape for a normal red cell. So this process shows that actually passing through the slit can facilitate the maturation of this red cell. And also we can correlate our some simulation result with some clinical finding, which in this 2001 blood paper, the author find that for the patient who has removed their spleen, their cell size is actually larger than the people who have their spleen intact. So this means that the spleen had the function of remove this cell size and the redundant surface area, which kind of is confirmed by our simulations. And also we can introduce some protein defects that are discovered in different blood disorders into this model and to simulate how the disease the red cell passing through this very narrow slit. So first, we introduced the defect discovered in the disease called Hereditary Spherocytosis. So in this disease, people find that there are band 3 defect and also spectrin defect in the red cell membrane. So in the band 3 defect, basically it means that the binding point at the center of the spectrum filament are missing. And in the spectrum filament defects, basically the band 3 contact point are intact. However, the density of the spectrum it become loose. So our simulation shows that in both cases of this defect, they actually can cause the vasculation of this red cell. Or vasculation from this red cell, and the red cell will continue to lose the surface area. And also we introduced another type of blood disease, a blood disorder. It's called elliptocytosis. So in this elliptocytosis, the protein defect occurred on the spectrum filament, where they will cause the disruption of the filament. So as we introduced earlier, we know that the spectrum filament network is responsible for the elasticity of the red cell. So after the spectrum filament is interrupted, so the red cell loses the mechanical property to recover the biconcave shape. So we can see that after the red cell passed through the slit, it cannot recover to this biconcave shape. And in a severe case of the disease, actually the passage of this slit can cause the lysis of the cell. Okay, so this is the end of the first part of this talk, where we show that by using this mechanistic model, we actually can use this computational model to explain some pathological phenomenon that discovered in the blood disease. So next, we change to another topic where we want to show that by using this blood flow model, we can predict some thrombosis induced by the COVID-19 virus. So this year we have moved into the third years of the pandemic. So everyone has get used to this new normal life with the virus. But if we turn our clock back to 3 years ago, if you remember that when the pandemic first outbreak, so the researcher first they discover or they build these interactive trackers to track the number of infections and hospitalizations at different scales, at the local and global scale. Basically, they can track the extent of this spread of the virus. And following that, so epidemiologists are starting to developing the mathematical model to use those numbers data to train the mathematical model and to make some prediction for the COVID pandemic. So this can help the policymakers to make some COVID-related restrictions, such as mask mandate, and also the lockdown of the local community or the entire city or even the country. Later on, big pharmaceutical companies like the Pfizer, J&J, and Moderna, they're actually developing vaccines and medications to prevent people from getting severely sick. And also, there are also biomedical researchers, including myself, are performing this mechanistic study to understand the pathogenesis and pathology of the disease induced by the COVID virus. And we hope that we can provide some insight to create a better treatment for patients with COVID-19. So the angle actually I try to tackle the COVID-19 induced disease is this macrothrombosis. So actually, since the COVID pandemic started, there are numerous clinical studies have shown that the COVID patients are at a high risk of suffering thrombotic events, such as venous thrombosis and arterial thrombosis. But in addition to those larger size thrombosis, those macrothrombosis, the autopsy examinations actually show that the macrovascular thrombosis, which means that the thrombosis in the very small macrovasculature, they actually occur in more than 80% cases of people who died of COVID-19. So in addition to the macrothrombosis occurred in the lung, the later studies show that the macrothrombi actually can be detected in other vital organs, such as the heart, kidney, and the liver. So all these findings indicate that this small-scale thrombosis actually could be the key driver or the main reason that caused the multi-organ failure in the severe cases of COVID-19. So at the same time, there are many prothrombotic factors has been reported that are related to the COVID-19. For example, such as the blastasis, which is mainly caused by the immobilizations of COVID-19 patients, and also the endothelial cell injury or inflammation. This is caused by the injury or death of endothelial cells due to the virus invasion. And also, people find that the COVID patients are always under this hypercoagulatable states, which is featured by the abnormal level of their coagulation factors, such as the increased level of factor 5, 8, 10 fibrinogen, and decreased amount of antithrombin, and so on. So another important feature of COVID-induced thrombosis is the involvement of the white cells, such as monocyte and neutrophil. So this is because the pro-inflammatory cytokine, the urea can stimulate the monocyte to synthesize the tissue factor, which is a key activator of the coagulation. And also they can release those tissue factor positive macroparticles into the circulation. So those macroparticles can actually bind to the activated platelets, neutrophils, and endothelial cells to initiate the thrombosis. And also, recently, a researcher find a number of different blood cell aggregates, such as the white cell aggregate, white cell platelet aggregate, and white cell red cell platelet aggregate in the blood sample of COVID-19. So those aggregates actually can block the small blood vessel, and also they can serve as a kind of seed to initiate the thrombus formation. So the motivation of my current study is that we want to quantify the connections between the multiple thrombotic factors reported in the literature and the extent of thrombus formation in the human system. By doing that, we hope that we can identify the key factor that mediate the process of thrombus formation, and hopefully we can provide insight to explore new targets for anti-thrombotic treatment. So here I want to emphasize the importance of a computational modeling in studying the COVID-induced thrombosis. Because of this infectious nature of SARS-CoV-2, so there is a limited access to the fresh COVID blood samples for in vitro experimental study. So we perform this computational predictive modeling is kind of very important to explore the underlying process of thrombus formation. So here is our model setup. So in our model, we apply a multi-physics and multi-scale computational framework that includes 4 modules. First, it can describe the hemodynamics and transport of coagulation factor. Second, it can simulate the blood cell mechanics, like the stiffness, deformability of the red cell, platelet, and white cell. And also, it can describe the platelet white cell adhesive dynamics and coagulation cascade reaction. So for the first 3 modules, it's actually integrated in the which is developed by our organizer of this complex flow seminar, Dr. Zhen Li. And in the 4th module, we actually can introduce different type of coagulation models into this framework. So in our current work, we introduce this very classic coagulation models developed by Dr. Annan at 2008. So in this model, it involves 23 species, which is described by 23 ordinary differential equations. And the key output of this coagulation model is the generation of thrombin, which plays the key role in activating the platelet. So here's our one case, our simulation. So in this case, we assume that there's an intersite in a circular channel, which it will express a tissue factor 7A complex on the endothelium cells. And this is going to initiate the coagulation and generate thrombin, which can activate the platelet. So you can see the passive platelet, which show up as the green. So it's going to become blue once they move into a boundary layer of the thrombin. And after the platelet get activated, they become adhesive, so they can adhere to the injured endothelium cells. Also, they can adhere to other activated platelet to form this platelet aggregate. And we actually can track or measure the growth rate of this aggregate by tracking the number of the adhered platelet. And then we feed this the growth curve with the exponential function to extract the growth rate of the platelet aggregate. So then we can measure the growth rate of the platelet aggregate at a different blood flow velocity, and it's triggered by different level of tissue factor 7A complex. So for example, here we can show that when the blood flow velocity increase, you can see that the growth rate of the platelet aggregate is first going up, and then they reach a maximum value, then start to decrease. So this is because when the blood flow rate is low, so there will be a limited number of platelets are delivered to the thrombogenic side, so the growth rate is limited. That is small number of platelets flowing in the blood. So once we increase the blood flow velocity, the growth rate will increase until they reach this maximum point, and then it starts to decrease. So this is because when the blood flow rate is increased, so the increased shear rate is going to limit the adhesion of the platelet to the thrombogenic side or the growing platelet aggregate. So that's why the high blood flow velocity is going to instead reduce the growth rate of the platelet aggregate. So this phenomenon is actually captured in a very old in vivo study in the mouse model developed performed by the Beckman and Born in 1970s. And after we play around with this blood velocity and different level of tissue factor 7A complex, we actually introduced the different level of coagulation factors from the patient with COVID-19. We want to examine which abnormal coagulation factors actually play the key role in triggering the thrombus formation. So we test fibrinogen, antithrombin, factor 8, 5, 10, and protein C. And then we all measure the growth rate of platelet aggregate. And we actually find that the reduced level of antithrombin in COVID patient actually is the key factor that triggers the excessive thrombus formation in the COVID-19 patient. And next, we actually introduced the Y cell into the simulation because the involvement of the Y cell in the thrombus formation is a key feature in the key feature of the COVID-induced thrombosis. So first, we assume that the Y cell is kind of play a passive role, so it just adhere to the thrombogenic side and without expressing any tissue factor 7A complex. So the coagulation is still initiated from the endothelium cell wall. So from our simulation, we actually see that by presence of itself, the Y cell is actually can promote the thrombus formation by altering the transport of this generated thrombin around the thrombogenic side. So the thrombin is actually highlighted by the yellow color boundary layer here. So it will trigger the platelet activation upstream of this Y cell and create platelet aggregate. And next case, we make the Y cell active. So this time the Y cell is going to express the tissue factor 7A complex itself and trigger the coagulation. So there will be no coagulation reaction on this on the vessel wall. And in this case, we also detect the thrombus formation and platelet activation. Actually, the platelets start to adhere to the Y cell. So we actually measure the growth rate for both cases, and we can see some different trend for the two activation mechanism for the thrombus formation. So when the thrombus formation is activated from the vessel wall, it actually is very sensitive to the blood flow velocity, which is shown by this black and red curve. You can see that when the blood flow velocity is increased, the growth rate quickly drops to 0. But if the thrombus activation is initiated from the Y cell, it actually is not very sensitive to the blood flow rate. So this study indicated that actually the activation of a Y cell in the COVID-induced thrombosis could be more dangerous because it actually can create some very persistent thrombus formation even the blood flow rate is high. So after we obtain this result, we actually contact our collaborator at MGH, and they also provide us some clinical data which show that there are different types of blood cell clusters actually are detected in the blood samples of COVID-19. So the typical blood samples, so the typical clusters include the platelet aggregate, erythrocyte aggregate, platelet leukocyte aggregate, and the leukocyte aggregate. So our collaborator also find that these different types of aggregate, they could be associated with the different clinical outcome of this COVID patient, such as they find that the leukocyte aggregate significantly correlate with the thrombotic complication experienced by the COVID patient. So they pose some questions. So how do you use your computational modeling to kind of explain the mechanisms underneath all this underneath this associations or correlation? So to answer those questions, we actually set up some new simulations. So first, we simulate a Y cell actually flowing toward existing platelet aggregate. So we find that when the blood flow velocity is relatively low, actually the Y cell can adhere to the thermogenic side and partially block the blood flow. And in the next case, we actually consider the interaction or the adhesion between the white cell and the red cell. This can be triggered by the generation of neutrophil extracellular traps, which functions as like spider net. Just can capture the flowing blood cell. So we can see that if we introduce the adhesion between the white cell and the red cell, so instead of just partially blocking the blood flow, they actually can cause a full occlusion of this microchannel. And in the next case, so we simulate the same simulation, we just increase the blood flow velocity and we see what's going to happen. And when the blood flow velocity is relatively high, we can see that this white cell is flowing through this platelet aggregate and forming this circulating red cell cluster, circulating white cell cluster. So this provides some mechanistic rationale for the observation of this white cell platelet aggregate observed from a COVID patient. And also, if we consider the white cell red cell adhesion, we can see that it started to form this white cell, red cell, and the platelet aggregate. And this type of aggregate is also observed from the blood samples of COVID patients. So in the next case, we simulate a flowing white cell moving toward the adhered white cell. So we can see this, the flowing white cell actually can push the adhered white cell and make it detached from the thermogenic side and forming this two white cell flowing cluster. And this also provides this explanation for why we observe this type of clusters in the blood samples of COVID-19. So to summarize the result of this interaction between the white cell and the platelet aggregate, we can see that it can largely depend on the blood flow velocity. So if the blood flow velocity is large, basically the flowing white cell will going to just interact with the platelet aggregate and grab some platelets and then forming this circulating cell cluster. But if the blood flow velocity is relatively low, it's going to adhere to the thermogenic side and forming a partial blockade or full blockade of the blood vessel, depending on the interaction between the different blood cells. And also we introduce this circulating cell cluster into a complex flow blood flow network. And we want to see how this circulating red cell impact the blood cell transport in this network. So this network we actually reconstructed based on the structure detected from the rat mesentery. And first we simulate the red cell only in this network. And we find that actually the red cell can be flowing into different branches, which means they can be perfused. All branches can be perfused. Then we introduce these two white cell clusters into this network. So we can see that this white cell cluster can actually pass through this bigger bifurcation, but eventually they are stuck at this smaller bifurcation and cause some congestion upstream of this bifurcation site. Similar things happen for the white cell and the red cell cluster. You can see similarly, they are stuck at the same bifurcation site due to the reduced deformability during the formation of this cell cluster. So by showing all this simulation result, we actually want to show that by using computational modeling, we actually can integrate different types of clinical data, such as the biochemistry measurement for the coagulation factors, and also the flow cytometry image data, and also we can connect those data with the final clinical outcome using the computational model. So to summarize on my talk today, I want to emphasize again the importance of computational modeling in the biomedical engineering by simulating some biological process that cannot be observed in vivo or in vitro, such as the red cell passing through the spleen process. I just introduced in the talk. And also it can be used to examine the hypothesis and derive new hypotheses and cross-validate the very important in vivo and in vitro experiment. And also we can dissect the process that involve many biological factors, such as the COVID cases, which involve many biological factors. We can quantify their contribution to the final thrombus formation. And that's all of my talk. At last, I want to thank our funding agency, NHLBI, for providing funding for our research, multi-fidelity and multi-scale modeling of the spleen function in sickle cell disease. And also, I want to thank our major collaborator, Dr. Ming Dao from MIT and Dr. Pierre Buffet from INTS Paris. Also, I want to thank all my collaborators at Crunch Group and also former Crunch Group members. And that's all. Thank you very much for your time and attention.
So okay, since Dr. Henry now is teaching in a class, so we cannot do Q&A. But if you have any question, you can email me, or I can ask to the question to Dr. Hini, or you can ask Dr. Hini directly. His email is on the CFSM website. Okay, thank you very much for your participation.
Hi.